Good morning, dear student friends. Welcome to online class again. In last class, we already discussed about the gene interaction. What is gene interaction and types of gene interaction. In that, allelic and non-allelic gene interactions. And one is allelic gene interaction. In that, two different example. One is law of incomplete dominance, and another one is the law of codominance. In last class, we already discussed about the incomplete dominance. Incomplete dominance, it refers to the dominant gene is expressive in nature in presence of any other gene. The gene which is a dominant will suppress another gene, what is called as a suppressive gene. In that the intermediate character is occurred. Example for the incomplete dominance is one is Mirabilis jalapa and another one is the dragon plant, snapdragon. And in this incomplete dominance, two parents, one is dominant and another one is the recessive. In that one is a red color flower bearing plant and another one is the white color flower bearing plant. Both of them are undergo to crossing to each other. The yield hybrid. In that the heterozygous pink colored plant which is occurred, which is an intermediate character in between the red and also the white, and which undergo to selfing, means the pink flower bearing plant which is a F1 generation is undergo to selfing, it is the F2 generation. In that F2 generation, the phenotypic and genotypic ratios are same, like 1:2:1. One is a red color flower which having homozygous and another two are heterozygous pink colored flower and one more is the homozygous white colored flower. So in allelic gene interaction there is a one more example, that is codominance. Let us study about the codominance here. This codominance, here both the alleles of gene express themselves in the heterozygotes. Phenotype of both parents appear in F1 hybrid rather than the intermediate phenotype. There is no intermediate character which is occurred by crossing of two parents which are having characteristic features of genotypes, like blood group A and blood group B. Here they are mentioned. The best example for this codominance is the blood group of human. In that, in human, AB blood group is the controlled by a single gene here. So of course the gene I represents the blood group. If I is consist of allele A, then it is called as a blood group A. If I is a gene which consists of the allele B, then it is a B blood group. In that I consist of both A and B, then the blood group is called as a AB. If there is no allele, means the small I, which represents the O blood group. And here only two alleles exist, A and B. Father with A blood group with the genotypes of capital A, and mother with B blood group, their genotypes is capital B and B, will have children with the AB blood group. Genotype is capital A and B. Both phenotypes are identifiable in the hybrid F2, segregates in the ratio of one is A blood group and another one is a 2AB and one more is the 1B blood group. Means it undergo to selfing, it cross, it releases, means IA and IB are crossed with each other. There is a codominance cross is represented in the corner of this slide, IA, IB which are crossed with each other. That is IA which represents the A blood group, IA and IB represents the AB blood group, IB again it represents the AB blood group, and IB represents the B blood group, which shows their genotypes like 1:2:1. One A blood group, two are AB blood group, and one is a B blood group. Means In out of 4 children, 2 of them are AB blood group and one is A blood group and another one is a B blood group. This is what it shows. If the two different alleles which are express their character equally, they are not, one is higher and another one is lower, in that there is no intermediate character which is occurred, and even though there is no law of dominance here, it is a kind of a dominance here, that is a codominance. Means capital A and capital B, if they are occurred in same genes, or same gene, so which express both equally. This is what showing of a codominance, which is also one of the allelic gene interaction. And second one is a non-allelic gene interaction. In a non-allelic gene interaction, the independent genes, that is non-homologous, the independent genes located on the same or on different chromosomes interact with one another for the expression of a single phenotypic trait of an organism. It means that the non-homologous chromosomes, the independent genes which are located on the same or different chromosomes, the independent genes located on the same or different chromosomes interact with one another. They are present on same chromosome or in a different chromosome of the independent genes. They are interact with one another for the expression of a single phenotypic trait of an organism, which is called as a non-allelic gene interaction here. There are 4 types of non-allelic gene interactions. One is complementary gene interaction, second one is a supplementary gene interaction, third one is the epistasis, and last one is the duplicating factor. But for your syllabus, only 3 of them. One is complementary gene interaction, second one is a supplementary gene interaction, and last one is the epistasis. In that there are the 2 types of epistasis we are supposed to study. One is a dominant epistasis and another one is the recessive epistasis. Let us study the first one is a complementary gene interaction. What is complementary gene interaction? See, it's very easy. Complementary gene interaction, the complementary genes, it may be defined as 2 or more dominant genes. Try to understand here. Complementary genes, it may be defined as 2 or more dominant, ಒಂದು ಅಥವಾ ಎರಡು dominant genes present on separate gene loci. They are present in a separate locus of the genes which interact to produce a particular phenotypic trait, but neither of them produce a particular trait in the absence of other. If in case in these 2 dominant, in this one is absent, which are shows there neither them produces the particular different character. The complementary genes were first studied by, there are 2 persons, one is a Bateson and another person is a Punnett. In the case of flower color of a kind of plant, what is called as a sweet pea, their scientific name is Lathyrus odoratus. It is a sweet pea plant. See, in the diagrammatic representation in the corner, we show the slide. One is white and another one is the purple color flower. See, their genotypes here, they are mentioned 2 white colored flower bearing plant. One is carries the genotypes like a capital C and small p, and another white flower bearing plant carries small c and capital P. So try to understand this one. In sweet pea, both the genes, I told you in complementary genes it may be defined as a 2 or more dominant genes. Dominant genes I am telling here, that is capital C and capital P are required to synthesize a kind of substance what is called as a anthocyanins. The substance is called as a anthocyanin. It is a kind of a pigment which causing the purple color flower. If the white color flower is there, in that it consists of the 2 dominant characters, like one is capital C and another one is the small p. They are present in separate plants, separate chromosomes, right? So those which are come in contact with each other, means during the time of crossing they brought together in a hybrid, both capital C and capital P are present in a single plant, which synthesize a substance in the form of a pigment what is called as anthocyanin, which brings the color of a purple in flower of the sweet pea plant. But absence of any one, absence of either it is a capital C or capital P, any one cannot produce anthocyanin, causing white flower. If in case there is no capital P, if there is no capital C, if in case only capital C is present, in that there is no capital P, then that flower is white, because of it is not stimulated to synthesize the anthocyanin. So only two genes which are required for the synthesis of anthocyanin, both of them are dominant genes like capital C, and another one is the capital P, remember. But in the absence of any one cannot produce the anthocyanin, causing white flower. So capital C and capital P are the complementary to each other. They are complementary to each other. In case the C is present, P is absent, means there is no production of a purple color flower. Hence, no, the C and P are act as capital C, capital P. I am telling here, capital C and capital P are act as a complementary to each other for anthocyanin formation. See, in this there are the two sweet pea plants bears one white color flowers, carries the capital C, capital C, and small p, and small c, capital P, brought together in a hybrid with the purple color flower and carries the capital C, capital P, small p. It is nothing but what? Heterozygous condition. The condition is called as a heterozygous here. Heterozygous genotypes in F1 generation. Once this F1 generation, see here, capital C, capital P, small p, which is shows the F1 generation. You know, F1 generation undergo to selfing or self crossing, which yield F2 generation. In this F2 generation, there are 16 plants are produced. So out of these 16 plants, 9 are purple flower plants and 7 are white flowered plants. Hence the phenotypic or in genotypic ratio is 9:7. The 9:7 is the ratio of complementary gene interaction. See in that the checkerboard which shows the white blocks are nothing but a white color flower, and purple blocks are nothing but a purple colored blocks. Right? The ratio is 9:7. 9 are purple flowers and 7 are white flowers. This is all about the complementary gene interaction. See, in the non-allelic gene interaction, there are the three types. One is complementary gene interaction, supplementary gene interaction, and epistasis. Now today we completed that complementary gene interaction. This is very, very important for the examination point of view. Please remember, and while you are answering, complementary gene interaction, all these are necessary. The points which are shown in the slide, all of them are very important. The checkerboard is also important and the explanation is also important. Thank you.